Okay, we start the last session this afternoon. And our speaker now is Professor Robert Gerlai, professor from the University of Toronto at Mississauga, the former president of the International Behavioral Science Society, distinguished investigator award from
IBEX.
And I forgot something else, the Vice President of
Research.
Industry.
And shall I put it up here? It's good? Okay, good. Thank you very much. It's truly an honor for me to be here for two reasons. Number one, I've never been to Israel. Number two, I'm honored to be among such great, talented colleagues who I admire. I'm not sure if I have anything new to say. So I'm going to go through a number of points which I put together, which I think Joan and Joan Kreb and other speakers this morning already covered. But I think it's important to emphasize these points again. And I did bring some new data for you to illustrate some of the points. And I decided to bring another species into the mix, zebrafish. So it's not going to be mouse data, it's going to be zebrafish data. And this is a test for you whether you're paying attention or not. Apparently only Wim Cruzio, who promised me not to sleep through my talk, noticed a typo in the title. It was actually a Freudian slip which exemplifies our efforts of trying to phenotype. So it's a pheno-trying. Anyway. So I've been interested in trying to understand the genetic and biological basis of behavior, and I think it's very important for us to phenotype, to characterize the phenotypical features, behavioral features of mutant mice. So, I changed my focus about 10, 15 years ago from mouse to zebrafish. Anyway, let me acknowledge the supporters, NIH and NSERC. NSERC is a Canadian funding agency, kind of similar to NSF, Basic Research. Some of these trivial points, I was expecting students in the audience too, so excuse me for the trivialities. But why should we measure behavior? Let me just start with that question. It's the ultimate output of the brain, but there are many practical considerations too. It allows us to detect functional changes in the brain, irrespective of neuron anatomical location or underlying mechanisms associated with these changes. So it's a tool that allows us to test mutation effects or drug effects. But it's also a clinical endpoint, and I've worked in pharmaceutical and biotechnology industry, and from that perspective, it's also very important to recognize that behavioral analysis is important. Patients and doctors don't care about the underlying mechanisms. It doesn't matter, what goes on inside the brain. What matters is whether the patient improves. And that's the other reason why behavioral analysis will always be an important question to address, whether you can measure the changes that drugs or whatever manipulations you use can achieve. How can we measure behavior? We covered this. There are many questions about this. The first we already discussed is whether we should use automated or observation-based methods, whether we should use nature-blind or nature-aware approaches, whether we should standardize the tests or use custom-made designs. Another point which has not been brought up, but it does come up in a serious manner in other discussions, serious because it's primarily grant funding agencies and referees almost always demand hypothesis-driven research as opposed to non-hypothesis-driven. But these non-hypothesis-driven studies in forward genetics are called unbiased screening. So I'm going to talk about that briefly also because we haven't mentioned that. And previously we also talked about whether we should use a single test versus a test battery. And last, if you screen for drug effects or mutation effects, should you use, what kind of strategies you should use to identify potentially interesting drug candidates or mutants. Top-down versus bottom-up test batteries. So this is basically what I'm going to talk about. And I will give you a short illustration of some of these points using a zebrafish example. All right, so pros and cons of observation-based approaches. Unlike often assumed in the literature, the human brain is still the best pattern detection device. So observation-based approaches should not be looked down on. A human observer can often see a lot more complicated posture patterns that even very complex computer vision-based algorithms will potentially miss. So I think it's an important pro. We should not discard observation-based approaches. It gives you flexibility if you are able to observe the animal. It gives you ideas what to look for. It's also inexpensive. So you don't have to have a complicated system to observe animal behavior. And if you have undergraduate students or technicians, it takes time, but it's cheap. Now, the automated behavioral approaches have also advantages. It allows you standardization and high throughput. But despite the automated feature and the seemingly improved precision, for example, we use video tracking systems. We have our own developed video tracking system and also use EtherVision, a standard Noldus company created tracking system. We find often really big errors in the tracking data that you actually have to manually observe or detect and remove. So the notion that automated systems are always more precise is not actually obvious and not always correct. But if you do it correctly, it can increase precision in behavioral parameters. And it can allow you to measure in a more quantitative ways certain things. For example, swim speed in our zebrafish, we can categorize it to slow and fast or medium speed using observation, but we cannot really measure the actual speed or the actual distance, precise distance from a particle set target point, etc. So there are some advantages. Higher cost, they are expensive. No matter what automated system you use, it is more expensive than just using an observer with a video camera or a stopwatch. So what I recommend in my lab is a healthy compromise, which I find it a healthy compromise. We always start with an observation-based approach, especially in a species that is new, like zebrafish, new at least to behavioral neuroscience, where we don't really know what to expect. It's always a good idea to start with observation and get some impressions, develop behavioral measures that you can eventually turn into an automated system measured variable. But I think it's true for also mouse or rat behavioral analysis as well, because although you know a lot about the behavior of these species, you don't know how the species will respond to a new mutation or how a new drug will affect its behavior. So you sometimes find unusual responses that you didn't expect or you would not expect if you just simply, you couldn't detect these responses if you were to use just an automated system. So it's always a good idea to take a look at things first and then develop an automated system based on the observation data. Came up before, we talked about ethologically relevant versus irrelevant approaches. Should we use nature blind or nature aware? So the argument is, we should be nature blind in the sense that we want to look for common features and mechanisms across multiple species. We shouldn't really worry about the species-specific aspects. We want to see the commonalities, especially if we are interested in translationally relevant aspects of brain function. However, subjects are tested in the lab in an artificial manner may not respond in an expected way, and this artificial aspect of the system, however you define artificial, is likely to induce a lot of error variation. So in that sense, it's a good idea to be sensitive to what kind of perceptual modalities, specific, visual or olfactory capabilities the species of your choice has, and what kind of motivational systems it possesses, what rewards or punishes the animal, and what kind of constraints, learning constraints it has. There are many species-specific features that you need to know before you design these tests. And again, for the mouse, they've been quite well worked out. For new systems like zebrafish, it's a big question. So we need to be aware of this. Again, it's a healthy compromise, which I suggest in my lab. We combine some aspects of nature-blind and nature-aware approaches. We take species-specific performance characteristics very seriously, and we combine these approaches with rigorous laboratory control to enhance replicability, which was outside, at least, of the traditional ethological approach, which used to emphasize observing animals in nature. Another topic which will be covered, I believe, by a following speaker, so I will zoom through this quickly. The pros and cons of standardization. The pros include the fact that if we standardize well-designed and well-defined set of tests conducted in a manner like a cooking recipe, following step-by-step what is written down, it's likely to give you results that are replicable not just within your laboratory but across. The problem is that it's never really described that way in the literature. So in fact, journals are against providing too much detail. And even tried and true behavioral tests that we call like Morris Water maze or plus maze, which we think are behavioral tests, are not well described. There are many, many variations, and Doug Walston, for example, conducted a review of how many different variables, environmental conditions vary across multiple Morris Water maze studies, and it's actually amazing how non-standardized these tests are, which is a mistake. Now, the other side of the coin, however, is if your particular behavioral test is so sensitive to small procedural or technical alterations, is this really a good, reliable test? Should we require a behavioral test to be more robust against these minor variations? So that's the point, the last point I want to raise here. Responses to a specific set of conditions versus robustness across a range of conditions. So ideally what you would like to have is a behavioral test that is fairly robust against these variations. Of course, the problem is, how do we know which test is robust against often unknown environmental condition changes that we are not aware of? And this is not something that academic professors struggle with only. It's a major problem in pharmaceutical research industry where a lot of scientists find positive, significant preclinical results, meaning drugs being efficacious, showing significant effects in animals, and then when you go to phase 2 human clinical trials, there is no significant effect. So the industry is struggling with this question, how is it possible to achieve better translational relevance using animals? And one obvious answer to this question, why it is often a failure, and 99.9% of drug candidates fail in phase 2, is because the human population is genetic variable. There's a ton of environmental variance in the population, and instead of mimicking that, what we use in the lab is a really strictly controlled environment where we reduce genetic variability and we reduce environmental variability too. So is it really a good thing for us to control all of these things? Or should we perhaps do the opposite? And it's a question. I don't really know the answer to it, but it's an important point. Custom design. We believe that standardization is an ideal solution, but is it really possible to standardize everything? And the answer to that is probably not, because there are many, many environmental factors that we are not even aware of that may influence behavior. But also, every time you test, for example, a new mutant or a new drug, your test that you believe is the Moritz water maze or the plus maze, no matter how well you standardize a test and how replicable the actual procedures are, your test is always an interaction between the actual procedure and the novel mutant in this case. So the outcome is not always easy to predict, and therefore, because of this interaction, it's every time you use a new manipulation, like a new mutation or searching for the effects of a new gene, transgenic mouse, the effects of a knockout gene, for example, using transgenic mice, you don't really know what to expect. So you think you are using a standardized test, but you may be missing out on being able to discover novel effects of your drug or the mutation. So therefore, custom designs are important. They may allow one to describe novel behavioral changes induced by these manipulations. So, but what should be the base of customization? And how should we compare the results across multiple labs? So that's the problem. So again, what I suggest, at least in our lab, is to again, some sort of compromise. Start with standard tests and observe possibly unique responses in these tests, and then form working hypotheses based on these observations, and then create custom-tailored follow-up analysis to prove or disprove these hypotheses. The other point I already listed up at the beginning is this hypothesis-driven idea. Grant funding agencies and referees love this notion. And indeed, you may get lucky based on past research. You may be able to identify a mechanism that is likely to underlie a behavioral change, a functional alteration that a drug or a mutation induced in the brain, and then you have a wonderful, nice story. But it's rare to be so lucky, and focusing on a particular hypothesis that you pick is often a premature decision, and overly narrow your focus, and it is not always a good idea. So instead, what in the screening field people suggest is what we call unbiased screening, which is the exact opposite of a hypothesis-driven analysis. In this case, the assumption is that there may be many mechanisms, many drug targets, many responses, many mutations that influence a particular brain function and behavior. And if you do an unbiased, thorough screening, you may be able to identify a large majority of these mechanistic details, as opposed to focusing on only a very narrowly defined subset of them. And this is especially true for behaviors that are interesting, because they are almost always complex, at least from a mechanistic viewpoint, like drug addiction, learning and memory, social behavior, a lot of neuropsychiatric disorders. They are very complex questions, and therefore focusing on a particular mechanism is almost always a bad idea. Now the problem is that really good high-throughput behavior screening tools either don't exist, or are too rudimentary, or are very time-consuming. So again, it's easy to say that we should have unbiased, thorough screening, but how exactly should we do this screening? That's a big question. And I think it's the last point, single versus multiple tests. Again, there are pros and cons. Single behavioral tests may be informative only if they have many built-in controls and perhaps multiple phases or parts, and they allow the recording of multiple behavioral parameters, which almost always aids interpretation. So we discussed this too. You don't want to just measure everything possible, but it's almost never possible to measure one behavioral response that corresponds to a complex psychological construct. And by that I mean, you cannot measure learning, for example. You can only measure learning performance in a learning task. Now, what does it mean? It's not just semantics, but if you just measure like escape latency to the platform in a Morse water maze, it is influenced by many, many performance features unrelated to learning and memory. And if you don't measure these additional behavioral parameters, swim speed, turning frequency, all kinds of swim path parameters, for example, then you may not be able to really interpret your the abnormality that you find in the reduced escape latency, for example, or increased escape latency. Now, test batteries take care of that by allowing you to measure many things in many different tests. If you organize them well, they may allow better interpretation of results. And, for example, dissociation of performance features, motivation, perception, motor function from the target behavior, learning, memory, or whatever you are interested in. I think this is the last point before the example and some general comments. Top-down versus bottom-up approaches. These are test batteries that are organized so that you can identify interesting drug candidates or mutants among many, many animals. Bottom-up test batteries are the ones that start with basic features of behavior. Can the animal see? Can the animal move normally? Is the animal motivated to perform a particular task? And then the assay goes on and builds on these findings and goes up more and more complicated and eventually get to the top of the complexity ladder. The advantage of this system is the systematic nature of the analysis. The disadvantage is that you have to run your animals through every single behavioral test, and every single animal, every single mutant, every single drug will have to run through this entire analysis. It's an enormous undertaking. The other approach is what I call the top-down approach, which starts with the most complex target phene, for example, learning or memory. And if you have a positive hit, that positive hit could be due to many things. Maybe the animal is impaired because it's unable to see the visual cue that is paired with the reward. Or maybe the animal is unable to move to the target platform, and therefore it's impaired in the task, not because it's unable to remember where the platform is, but because of a performance impairment. But once you have an identified mutant in a forward genetic screen, then you can run follow-up studies to tease out what may be behind this. So the advantage of this kind of top-down approach is that you're not wasting time on testing every single mutant. You are focusing on the mutant that already gave you a positive hit, and then you can discard those that are not really interesting from your viewpoint. And if you're looking for a mutation that affects learning, you will discard those mutants that were impaired only because of a performance impairment. But at the end, you will get to a few perhaps that are completely normal in terms of performance characteristics, but it still shows impairment in the learning task. So it's a bit more efficient. Now, last few comments before I show you an example, actual data set. We all complain about this. that behavior is too easy to measure. The problem with that is that a lot of scientists who are not behavioral experts can easily generate data by throwing the mouse in a large pool of water and having a stopwatch. And as you all know, it's not so simple. You really need to worry about all the things that we already talked about and the things that we are going to talk about. So it's just an apparent ease. In reality, it's a very, every behavioral task is a complex set of characteristics, and it's a very difficult task to run it exactly the same way. So that's why behavioral tests are variable and finicky. And that's why behavioral analysis is a double-edged sword. It's seemingly very simple, but in reality, it's a very complicated endeavor. So the apparent methodological simplicity masks the complexities of conducting behavioral analysis. Now, the other point I want to make, and the example we showed this, that we look at behavioral variability or the problem with replicability as an issue, and it is. But as Joan already mentioned, it may also be viewed as an opportunity. Behavior is very sensitive by design, so to speak, to all kinds of environmental influences and to many, many genes. So this variability actually represents an opportunity to discover how the brain works. And it allows you to discover all kinds of environmental effects and all kinds of intrinsic factors that alter brain function. So it is an opportunity for us. Now, the last point I want to emphasize that we have not talked about is the choice of the species. We all work on mice. I used to work on mice and rats as well. But I want to make a point that adding more species to the mix will actually perhaps increase our ability to become more replicable, to find scientific replication, not necessarily, replication of results. I don't expect zebrafish to behave exactly the same way as the mouse in a water maze, for example. But in terms of scientific conceptual aspects, adding new species to this mix will aid our understanding of behavioral and brain function-related phenomena. And the reason is this. It's a kind of a simplified version of what I explained in a publication, but if you consider, if you accept the theory of evolution and the fact that all species are related to each other, if you consider 3 species in here, what you're seeing here is the Boolean algebra showing the set of mechanisms that underlie a particular function, in this case a behavioral function in species A, species B, species C. So if you have 2 species, you have a particle overlap, and you have a non-overlapping area between A and B. If you have 3 species, the likelihood is that there will be a smaller overlapping area among all 3. Now, this particular area represents, some argue, the core mechanisms that perhaps evolved upon which layers and layers of additional mechanisms were built by evolution, simply because evolution cannot really start from a fresh design. It only can modify existing features. So this layering is a general phenomenon if you look at any kind of characteristics, behavior and brain function included, which is exemplified by this. So, the newer the species, these are the evolutionary youngest set of mechanisms, and this is the oldest, most ancient. If you combine species, multiple species, and you analyze their mechanisms, the common overlapping area perhaps is the most interesting, and this would allow you to be the most translational relevant, because you have already discarded this area which may not be as important for the particular function. So that's one of the reasons why I recommend using, for example, zebrafish. But there are many other particular reasons why we chose this fish. And I'm going to present an example today on a fetal alcohol spectrum disorder model we are developing in our lab with this little fish. It's a tiny creature, 4 centimeters long when adult. They're very cute, easy to keep in large numbers. We have a small, facility about like 4 rooms like this corner, and we can house 60,000 fish, basically a small town's worth of human population in this few square meters of lab space. So it's very efficient and cheap. Just briefly, you're not alcohol researchers, but alcohol consumption during pregnancy is an enormous medical problem. It's the single most frequent cause of preventable mental retardation. So, and 1 in 100 newborn will be born with some form of fetal alcohol spectrum disorder. Particularly in the milder forms of the disease are very prevalent. And there are many behavioral abnormalities that will stay with throughout the life of these children as they grow up. They will never really, they will always suffer from this. So for example, increased anxiety, aggression, abnormal sexual behavior, and very importantly, abnormal social skills, social behavior. Now, you would say, well, how can you model this complex behavior using zebrafish? Is it relevant to use zebrafish for this kind of disease perspective, disease reason? Can you discover anything? And the answer is yes, because if you find a gene that is involved in a particular function in zebrafish, the nucleotide sequence homology with human genes is really high. So if you find a zebrafish gene involved in a particular function, very likely you will be able to identify a human gene, a homologous human gene that will serve a very similar function. So zebrafish is a discovery tool, a hypothesis, mechanism hypothesis generating tool that will make discovery of genes and biological mechanisms involved in particular diseases, human diseases, much more efficient. That's the argument at least many of us make. Very complicated slide, just to impress you with the fact that zebrafish has a complex behavioral repertoire. Many mouse tests that are quite well described and used in the mouse literature have homologs or similar tests available in zebrafish already. And with that, let me just talk briefly about our fetal alcohol spectrum disorder model. One reason why we chose zebrafish is very easy to administer alcohol to zebrafish. Not exactly like this, but you can put zebrafish into the alcohol solution, adults and the embryos. They swim in alcohol, or in this case the eggs, and 24-hour post-fertilization where we use the alcohol manipulation. These fish are inside the egg. You put them into the alcohol solution. Alcohol diffuses into the egg through the eggshell, and the embryo is exposed precisely at the given developmental stage for a period of time that you set, so you have complete control over when and how much and how long alcohol gets to the embryo, as opposed to in the mammalian system where it's very complicated. The maternal physiology makes it really complicated to control. Another point is that zebrafish have a complex behavioral repertoire, including social behavior that we can measure. 5 minutes, okay. Right. So this is the assay that we developed, a very simple tool with which we can measure responses. We tested many things in zebrafish, and I'm going to show you this test because this showed us a really interesting result, a change in behavior induced by embryonic alcohol exposure. So what you're looking at here is a single fish put in a tank. It's about a 50 centimeter long tank. On each side of the tank there is a video screen that can present an animated image of a shoal. So these are moving zebrafish that look like zebrafish, and we have shown that subjects like this guy will treat these images like live, real zebrafish. What does it mean? Before you turn on the images, the zebrafish subjects are about 25 centimeters away from the image screen, which is basically, on average, they're in the middle of the tank. When you turn on the images, it's like a glue. These fish, like a magnet, are drawn close to the image side, either this side or that side. And as long as the images are shown, the fish are staying close to the image screen. When you turn off the images, they go back to about the middle of the tank. Very robust behavior, very easy to induce in an automated manner, and it's easy to measure in an automated manner too. We use computers to turn on the image. It's all computerized. We developed the software application that allows us to control the speed, the size, the location of these images, etc., etc. And we use a tracking system that measures exactly where the experimental fish is. So this is what we found. First, we couldn't believe this because it was too good to be true. But what you're looking at is adult zebrafish, fully grown, adult, sexually mature, 4 centimeter long zebrafish that look normal, swim normally, but were exposed to alcohol at their 24th hour post-fertilization stage in the embryo. So this is the 0% control. This is the 0.25% alcohol group, 0.5, 0.75, 1% exposed, 1% outside concentration. Inside the egg, it's 1/30th of that. So it's a very small amount of alcohol that actually reaches the brain of these embryos. And what you see is an almost perfect linear dose response curve. So this is the change from here to here. So the difference between the pre-stimulus and during stimulus response. So you see this beautiful reduction of the reduction of distance. So basically at 1%, these fish are not responding. This is not significantly different from random chance. So 1% abolishes the shoaling response. Now, there are many, many problems with this. It could be vision. Maybe these fish are just not able to see the images. Maybe they're not able to move to the images. We excluded all of these explanations. We know they can see how. I don't have the data and time to explain, but we have another behavior measure that shows that these fish respond to the images just normally, but not by moving closer to them. They slow down. But their swimming speed in general is normal. All fish slow down the same way. And all fish swim with the same speed before the images and slow down in response to the images, irrespective of the alcohol dose. So they do see the images. They just choose not to swim closer to the images. Now, the other independent replication is a completely different test. In this case, what you're looking at is a live shoal that we videotaped from above. Each shoal member, 10 fish, are of the same alcohol treatment group at 15 days of age, 40 days of age, 70 and 91 days of age. Control to 1% exposed. So if you look at the controls, you see the development of shoaling. The shoal cohesion increases. The group cohesion is strengthened. But if you treat these fish with alcohol, you see a significant impairment. Not as nicely dose dependent, but at least you do see a significant impairment in the alcohol groups. So here vision plays no role, or at least vision alone is not the only modality that allows the fish to shoal closely to each other, because they use the lateral line. If you blind the fish, they will still shoal. So we can exclude vision here as a factor that influences the behavior. And it's an entirely different setup, but the same finding. So this is so far so good. Now, for some reason, I don't have... yeah, okay, good. So what does the reduced response to conspecific mean? Could it be due to abnormal motor function, abnormal vision, abnormal motivation? And what are the underlying mechanisms? This is what I already mentioned. I didn't remember I had this data. This is the actual distance moved before and during the image presentation. And there is no difference here. So all fish respond with a reduction of speed, swimming speed. And therefore, we concluded that embryonic alcohol treatment disrupts shoaling induced by conspecifics or their animated images in the adult, and the disruption is not likely to be explained by changes in visual or motor function. But we got a surprise. After we found this, we could not replicate it anymore. In another study which used the exact same alcohol doses, same alcohol exposure procedure, same behavioral test paradigm, we failed to show the alcohol-induced impairment in shoaling responses in these fish. And here comes the opportunity. We looked at everything we did, and we discovered that the rearing conditions were different in this study. We discovered that we reared these fish in a larger tank as opposed to the original study in a smaller tank, and in a lower density as opposed to the high density in the original study. So it's a very interesting finding. We don't have conclusive systematic analysis of this and conclusive evidence for what exactly made a difference. But this surprise has given us a lot of working hypotheses that we can follow up on. And the reason why it's actually very interesting is that we never really wanted to generate an FASD model that is so robust that others have done before, that half of the head is missing in the fish, and no matter what you do, it's always like that. What we found here is fish that whose behavior is slightly impaired, but everything else is normal, and you could actually apparently rescue that behavior by an environmental manipulation that we were not aware of before. Some sort of enrichment-induced rescue is happening here, and now we are in a process of discovering what is that enrichment and what may be the mechanism of that enrichment that allows us to repair or modify the developmental trajectory of the brain in a way that the behavior doesn't manifest anymore in the adult. So that's kind of a nice thing. So I think, again, just to go back to the original point that others have made already, variability and lack of replicability sometimes does represent opportunity that you can utilize and discover. You can utilize for discovering functionally important changes in the brain. I don't know if I have like 1 minute, just briefly about the neurobiological mechanism. If no, okay, good. So I stop here. Okay.